So, good evening. I am Dr. Vishwas Tripathi, and I welcome you all again in this webinar on therapeutic approaches in targeting COVID-19 pandemic, drug repurposing, and vaccination. We are really sorry that because of some technical problem in the first sessions, many of you cannot join. But actually there was a limit of 100 people and we were not aware of that as we had got huge response. So we are conducting it the second time, the same thing, the same presentation. We are continuing with that. So let's start with, one more thing I would like to say that since there's a limited time of 40 minutes for this presentation, so I will request you all that the question answers will be taken after the presentation. So please don't communicate in between, else it will disturb the whole presentation. Thank you very much. So our whole discussion will be confined in four major parts. In the first part, I will be discussing about the COVID-19, how it arose, how the SARS-CoV-2 was named, the different aspects of the virus. And in the second, we'll be discussing about its stat part. In the third part, I'll be discussing the virus origin, its transmission, how its replication cycle goes on, how it affects different parts of our body, and how there's a systemic immune response, what we call as cytokine storm. In the fourth part, we'll be focusing on the main part of this presentation, that is therapeutic approaches. In that therapeutic approach, I will be discussing, I will be mainly confining this discussion to drug repurposing and vaccination, but we will be discussing all the possible options of therapeutics which can be utilized in the treatment of COVID-19. So as we all know that this COVID-19, that is coronavirus disease 19, is caused by the recently emerged virus SARS-CoV-2, severe acute respiratory syndrome coronavirus 2. It has been named CoV-2 just to differentiate with the previous one, which had also emerged in China in 2003. Now these SARS-CoV-2 viruses, basically they are single-strand RNA beta coronaviruses. Beta coronaviruses are the viruses which are having zoonotic origin, that is they have emerged in the animals. Before this China episode, I mean before this December 2019, there was no report about this virus. So whatever information we are getting, there are so many speculations, hypotheses. So whatever science is surfacing, they are not still validated. The first case was reported in Wuhan, China. As we all know, in December 31st, the virus has affected around more than 190 countries, that it has affected across the globe, and the virus has not only affected medically, but it has affected socially, politically, economically. Hardly any aspect of the life is left which has not been touched by the virus. Nobody has imagined that the whole world would be in the lockdown for such a long duration. Now it has been declared as a pandemic. One interesting fact with this virus, which can tell us easily that how fast the transmission rate of this virus is, that within 45 days the virus turned from epidemic to pandemic, and it was declared on 11th March by WHO as pandemic. And of course the scientists, the clinical researchers like us, we all are trying to solve this mystery of COVID-19. So symptoms of COVID-19, we all know that the virus has the common symptoms of the virus is high fever, tiredness, hemoptysis, dry cough, pneumonia, difficulty in breathing, renal failure. But among these symptoms, there are some symptoms which are very common. So it has been observed, as per one report published in NEJ, among all these symptoms, high fever has been reported in around 86% cases, and dry cough has been reported around 68 to 70% cases. Similarly, the tiredness, myalgia, when we feel fatigue, we feel too tired to work. So all these tiredness symptoms, fatigue symptoms have been reported around in 40% cases. So these three symptoms are the primary symptoms which are common. Apart from them, there are several other symptoms which has been observed in some cases, like uh, in some cases people may have hemoptysis, that they would be coughing blood. Pneumonia, the ultimate stage is that the lungs get damaged, and because of the injury in the lungs, the pneumonia occurs. That means that infection then gets transmitted into the blood, then that leads to the septic shock, and finally it goes to the renal part, and then the kidney gets damaged. There are so many systemic response. This all we will be discussing in detail. Now, I would not discuss much about the preventive measures, because we are listening this everywhere on the newspapers, in electronic media, in social media, even our phone call is telling us this. So you all know that we should wash our hands, keep a distance, try our best not to touch our eyes, and so many things. These are the things which are very common, everybody knows. Now, stats about the coronavirus. We have data till April 19th, and the active cases were found till date were somewhere around 17 lakhs, 16 lakhs 91,000. We can roughly say it's somewhere 17 lakhs. And 97% of those cases were of mild nature, and 3% of the cases were of serious or critical nature. Now, origin, transmission, and characteristics of this virus. This is a very beautiful presentation. This presentation will give you a holistic picture of how SARS-CoV arose, what are the different structural proteins, non-structural proteins, what are the different structural features of the virus. So everything is given in this presentation. This is very beautiful presentation. So that's why I have chosen it. Here you can see that the viral factors, from where they came, this is still a matter of debate. It is still controversial. China has a different version, rest of the world has different version, but there are so many things which scientifically also indicate that this just can't be a coincidence. Earlier, the story came with that the virus arose from snake, then the story of bat came into picture, and recently we are having pangolin origin of this virus. There's one hypothesis that says that the virus originated from pangolin because of the similarity with the receptor binding domain of pangolin SARS with this novel coronavirus. So you can see the structural part of the protein. Among the structural part, the part which is very important is S protein, spike protein, because this is the protein which first comes into the picture. This protein binds with the ACE2 receptors present on the host. The S protein is having two subunit, S1 and S2. And S1 subunit decides the tropism of the virus. We say tropism, that the virus, which line of immune system will be managed by the virus. Similar case of tropism we have in case of HIV also, whether I am tropic or D-tropic, and there's so many things. So S2, there is another spike protein S2. This S2 protein, basically there are HR1 and HR2 heptide repeats. These HR1, HR2, they together form 6 helical bundles, and those 6 helical bundles help these spike protein to fuse with the host membrane protein. So this helps the in the fusion, and it helps the virus to come in close proximity with the cell membrane. Thereafter, we have another M protein, that is membrane protein. We have non-structural proteins. Non-structural proteins are those proteins which are not which doesn't participate in the structural part of the virus. Then we have accessory proteins. Accessory protein have their regulatory role. Now the host factor. Among the host factor, the most important one is angiotensin converting enzyme, ACE2. Now this ACE2 receptor is present on nasopharyngeal tract, it is present on intestinal GI tract, it is present on lung alveoli, it is present on kidney, and recent reports say that a large number of ACE2 receptors are also present on the on brain cells. So these ACE2 receptors are the receptor with which the virus binds. And one important thing which we all would be interested in knowing, that if the virus has similarity with the SARS and MERS, the earlier evolved viruses, uh, so why we are so much panic about this SARS-CoV-2? The answer is that the SARS-CoV-2 receptor binding domain of spike protein is having very high affinity for the ACE2 in comparison to the previously emerged viruses. Now we all know that the most vulnerable population is people who are of old age, people who are above 65 years. So all those people uh, so all those people and especially the people those who are having pre-existing conditions, like they are having diabetes, hypertension, cardiovascular problem, COPD, or any kind of asthmatic problem, special kind of allergy, all these people are very prone to this disease, even among the elderly. And the virus leads to the, here you can see the cytopathic effect. Cytopathic effect is the effect which has been caused because of the virus infection in a cell. So when virus infects a cell, there are so many structural changes that takes place in a cell, and together we say this cytopathic effect. Then there is another term here, cytokine storm. Cytokine storm is a very important phenomenon, at least in this case. In fact, the cytokine storm and the cytokine release syndrome are severe inflammatory responses. These are all three the interchangeable terms, the same terms. So this cytokine storm has been observed in case of several respiratory viruses. The most common one is influenza, and the same problem here in case of SARS-CoV-2, also in case of COVID-19 we are observing. And the recent hypothesis, the recent findings suggest that the main reason, one of the main reason of the COVID-19 patient's death is this cytokine storm. What happens when the virus infects the alveolar cells? There is too much of cytokines which are poured in one go, and this cascade goes on in a very severe and quick manner that leads to a systemic response that lead to organ damage and so on and so forth. Then the situation gradually worsens. So the people, I have already discussed these things. Yeah, respiratory distress syndrome. What is respiratory distress syndrome? We say it acute respiratory distress syndrome also, ARDS. When in the air sacs of the lung, the fluid gets deposited, so surfactants are reduced. And the surfactants are the protein which are present on the type 2 cells of the alveoli. These surfactants are basically, they assist lung to inflate and they ease our respiration process. But since the surfactant is lost and there is a fluid deposition, so that basically leads to the compromise with the oxygen conduction, the gaseous exchange of oxygen to other organs. So basically it leads to the oxygen deprivation, and that is why there is acute respiratory distress syndrome. The patient feels very uncomfortable while respiring. During respiration. Septic shock. Yes, septic shock is another after effect of the COVID-19 infection. In septic shock, what happens when the infection becomes very severe and it travels across the body, the whole part of the body, and then the infection becomes systemic. So that is known as septic shock. The after effect of the septic shock in terms of symptoms is that the patient's blood pressure goes very down, low BP is there, the patient's arms and legs, they become very cool. So these are the symptoms of that. Another one is coagulation dysfunction. Coagulation dysfunction, basically thrombocytopenia has been observed in these patients. Now those who are not from the biology background, for them I would like to tell that thrombocytopenia is nothing. We were discussing coagulation dysfunction, the thrombocytopenia. Now that these thrombocytopenia, cytokine storm, they all lead to multiple organ failure. Now, this is a diagrammatic representation of how the COVID-19, I mean the SARS-CoV-2 virus infects the host cell, the virus life cycle. So here you can see, as I have already discussed to you all, that the virus spike protein plays a very crucial role. The spike protein binds to the ACE2 receptor. Here, one interesting fact which yesterday I had gone through, I would like to share with you all, that the ACE2 receptors, they bind to the virus and they have high affinity. But some rumors in the social media and electronic media are circulating that the patients, those who are taking anti-hypertensive drugs, I mean in a layman's language, the patients which are taking the blood pressure medicines, so those patients, they are vulnerable to this kind of infection because their medication increases the expression of ACE2 receptor, and that leads to the that may further make them prone to this infection. So let me clarify you, till date we don't have any finding of this type. We can't say that it is because of olmestron or amlodipine somebody is taking and his ACE2 receptors are increasing and then the person is becoming vulnerable to the COVID-19 infection. So those who are taking these medications, they must continue with their medications as per their doctor's prescription. Now, the after that, the virus fuses. As I told you, HR1 and HR2, these two proteins play a very crucial into the fusion, and thereafter uncoating. Then the virus protein gets translated, and through rough endoplasmic reticulum, you know that we all know that there is GRL system in the cell. So it assembles, there's a wording, and then ERGIC, endoplasmic reticulum Golgi intermediate complex, through which it goes to Golgi body. There are glycosylation and the post-translational modifications, and then in the vesicles it is exocytosed and the virions are released in the surrounding media, and then they infect the other cells. Now this this is a presentation which will tell you how the COVID-19 affects our body. So very simple presentation. You can see here that they have given you a picture of lung, and the alveoli region is here enlarged. So the alveoli, we have different cases of infected, moderate, and severely infected patients. In case of the normal one, you can say the healthy one, the gaseous exchange is normal. In infected, it has been shown that in the air sacs there are SARS-CoV-2 is present. Those structures have been enlarged. In the moderate cases also they have shown vasodilation has been taken place. Macrophages have been recruited because of the virus infection. And here you can see, yes, yeah, the fluid filled interstitium. That means the fluid has come to the air sacs, and loss of surfactants. Surfactants are very important secretions, the outer membrane secretion of the type 2 cells. Our alveoli is having our alveoli are having two types of cell, type 1 and type 2. So the type 2 cells basically are those cells which secrete the surfactants, and these surfactants, they reduce the tension of the lung, and because of the reduction in the tension of the lungs are easy to inflate. So your nasopharyngeal tract can easily you can easily breathe because without any interruption. But what is happening here, since the surfactants are lost because of this inflammation, so this leads to respiratory syndrome, acute respiratory distress syndrome, in which the lungs are compromised and finally the alveoli, they get damaged. Now, how our immune system fights with the SARS-CoV-2. So after infection, type 2 cells, as I told you that the type 2 cells are those cells which are present in the alveoli. So these type 2 cells, they release inflammatory signals because they are the cells which express ACE2 receptors. They take up the virus infection. So of course, it is a natural process of our immune system that any infection would be there. So they'll go through the inflammatory, they will raise the inflammatory signals. Those inflammatory signals will further recruit the macrophages. They will release their cytokines. Subsequently, neutrophils will also be recruited at the site of infection. Those neutrophils, they are supposed to release ROS, reactive oxygen species, to destroy the infected cells. Type 1 and type 2 cells, uh, type 1 cells and type 2 cells, both are present in the alveoli of our lung, are destroyed, followed by collapse of the alveolus. And when there is a collapse of the alveolus, that means surfactants are lost, the boundaries are breached, and then there is a liquid accumulation. That liquid accumulation finally leads to the acute respiratory distress syndrome. Now, if inflammation becomes very serious, very severe, so in that case, it becomes systemic. Systemic means when it affects your whole body, your whole systems. So systemic inflammatory response syndrome is there, that finally leads to the, since the infection then goes to the blood, then it leads to the septic shock, low BP, and multi-organ failure. Yeah, one important finding is cytokine storm. I had already discussed it. Cytokine storm, as I told you, that recent findings suggest that the COVID-19 patients' death is fueled by this cytokine storm phenomenon. And this is not only in case with the COVID-19, rather in most of the respiratory viral pneumonia infections, especially in case of influenza, also we observe the same kind of phenomenon. Now, this is one diagrammatic representation which is showing how our immune system works. So here you can see that the delayed or suppressed type 1 IFN response during initial infection. Viral replication triggers hyper inflammatory conditions, and those hyperinflammatory conditions lead to the cytokine storm, influx of activated neutrophils, inflammatory monocytes, macrophages, and then they subsequently release their cytokines, and this cascade goes on. Then finally it leads to a very severe kind of inflammatory response. Th1 Th17 cells, Th1 Th17 cells are nothing but they are the cells of Th lineage. Th1 and Th17 cells are pro-inflammatory cells. Th17 name has been given because it releases IL-17, interleukin 17. So that is why it has been given. And this IL-17 secretion has been linked with the IL-6 pro-inflammatory response. Now let's discuss the therapeutic approaches. What all therapeutic options we have in case of COVID-19. So these are all the possible options which I'm going to discuss in one slide. But after that, we'll be confining our discussion to only drug repurposing and vaccination. And I'll be telling you why I have selected only these two things. So the first option is that anybody, a layman even can think of that why don't we design a vaccine? So let me tell you that the designing of the vaccine, at least in case of viruses, is a very tedious, cumbersome process, a very time-taking process. Huge amount of money is involved. It has to go through several regulatory steps, and then only a vaccine comes to the market. Another important fact which I must share with you all, that the flu vaccines, the success rate of these vaccines is very, very low. From 2003 to 2016, the vaccines which were tried for different kind of respiratory virus related infections or the flu related infections, their success rate was only and only 16%. You can think of how risky and how challenging it is. Now, as per the WHO advice, even if we consider the urgency, the medical urgency situation of COVID-19, we will be having a safe and effective vaccine at least in 18 months. It will take minimum 18 months. Some other agencies, those who are working very fast, some pharmaceuticals are also involved in that. They are saying that we can give vaccine as early as in the early 2021. But we can't wait for that much time. And moreover, even the vaccines which are going through the ongoing clinical trial, even in those cases, the CIP, one of the very renowned organization which manages funding, and it cooperates with the WHO in designing of the vaccines, their formulation, their strategy, all regulatory steps. Even they say that the potential rate of success of COVID-19 vaccine is around 10%. So still the vaccine part per se will not solve the problem, and moreover we have to wait for a longer duration. So we can't afford that. Now another option is monoclonal antibodies. Yes, well, in case of several viral infections, monoclonal antibodies have been used, and they are used as a neutralizing antibodies. It's a good idea, but I will say, being an immunologist, that monoclonal antibodies have their own problem. First of all, the main problem is the cost. It won't be accessible to each and every person, the cost involved in that. Second problem is that managing the pharmacokinetic parameters of the monoclonal antibodies is a difficult task. Third problem is, in certain respiratory viruses itself, it has been observed that these antibodies, they itself act like a ligand, and then they transduce several other pathways. In some cases, they show very severe kind of hypersensitive response, undesirable immune response. So these are all limitations associated with the monoclonal antibodies. Moreover, monoclonal antibodies are also not a ready-made or immediate option for COVID-19 treatment, which we require as of now. Now another one is oligonucleotide-based therapies. Yes, oligonucleotide-based therapies are also emerging as a very promising therapeutic option, but that will also take a lot of time, because it has to go through you have to identify those oligonucleotides, then you have to go through in vitro animal models, then clinical trials, different regulatory checks of FDA, and finally it will get not. So all this process is a time consuming process. Yes, the next one is repurposed drugs. I must say this, that is why I have selected drug repurposing in this topic, that repurposed drugs or drug repurposing is the only and only immediate solution of this problem, because in this case the drugs are time tested and several regulatory barriers we can escape. So for considering the medical emergency problem of COVID-19, this is the immediate solution. I'll be discussing it in detail later on in subsequent slides. Now peptides are also emerging as a therapeutic option, but peptides have their own problem. First, the two main problems that peptides face as a therapeutic option is that chances of their proteolytic cleavage, and the second one is their short half-life. So managing that Tmax or the pharmacokinetic parameters is difficult. Interferon therapies, yes, interferon therapies are also used, and in certain viral infections cases it has been very successful. But interferon therapies have also their own limitations, because managing that effective dose level to the final site is a difficult task. And second one, sometimes they also show systemic inflammatory responses, so they show undesirable immune responses. Small molecule drugs, yes, we can work on that. Several people are working on that. People are working on the small molecules which have been approved by the FDA, already approved. People are exploring new hits. So in that case, what you have to do, you have to do a lot of structural analysis work. Then you will identify the leads. You will then identify your hits. After that, you will go for in vitro studies, then animal models, then followed by clinical trials. Again, this is a time consuming process, and the success rate is very low. So keeping everything in mind, we have only two solutions which I personally feel that drug repurposing is the immediate solution. And if we can design vaccines, and if the vaccines get through all those barriers, so that would be another fantastic approach, at least for the preventive purpose. Now, what could be the possible lead in treating COVID-19? Very interesting. So as we all know that the SARS-CoV-2 is very, very new to the human population. We don't have any report on that, and that is why so many false positive results are coming. So many facts are surfacing each and every day. So from where should we get the lead? So we can get the lead from the previous related versions of the virus, not exactly the same, but we can take the lead from the SARS and MERS. SARS, which arose in China in 2003, severe acute respiratory syndrome. You can say it's SARS-1, and the other one is Middle East respiratory syndrome. So we can get a clue from that. We can utilize all the experiences, the lessons, the leads, the research findings we are having from these two viruses, and then the same can be expanded in case of SARS-CoV-2. Another important thing we can do is that uh, we can repurpose all those drugs which have already crossed the clinical trial barriers for the treatment of SARS and MERS. The same can be explored for repurposing in case of SARS-CoV-2. Now, what the therapeutics options we have? So mainly the therapeutic options are, if I divide them in two major parts. So one, the first option is in which we can target the viral agents. So these viral agents, virally targeted agents, they include non-structural protein and structural proteins. I must clarify it here that the proteins which I have shown here are not the only protein which are present in the virus. I have written here only those proteins which may act as a good candidate or which have been reported as an attractive candidate in targeting this virus. So we have non-structural protein, 3-chymotrypsin-like protease, what we say as 3CL protease is very common, papain-like proteases, helicases, and RNA-dependent RNA polymerase. These proteins can be targeted, and structural protein, spike protein, as I told you, that this is the protein which is the first protein which interacts with the host cell receptor ACE2. And if the another target option could be the host targeted agents, so we can target the host proteins. So ACE2, which is present on alveolar cells, type 1, type 2 cells, on kidney cells. So this ACE2, the virus first binds to the ACE2 receptor, so we can target the ACE2 receptors. TMPRSS2, basically they are serine proteases, and they can cleave and activate the spike protein. So they help in the cleavage of a spike protein. So if we can inhibit these serine proteases, that could also be a therapeutic option. Now coming to the drug repurposing. So drug repurposing is nothing but basically drug repurposing is simply using already marketed drugs for other indications. Let me give you one example. Suppose methotrexate is a drug which was originally discovered for cancer treatment. But now most of us, if we know methotrexate, so we know it because of its use in the rheumatoid arthritis treatment. Similarly, metformin, which is emerging as a very promising repurposed drug in case of metastatic cancer treatment. Metformin is an anti-diabetic drug. We all use in those who are suffering from diabetes, they take metformin or metformin in combination with other gliptin salts. So metformin is primarily, it was discovered for the treatment of diabetes, but now it has been repurposed for now people are exploring it to repurpose for metastatic cancer. So this is drug repurposing. That's why the term is repurposing. We are again repurposing that drug. So what are the advantages of the drug repurposing? So the most important advantage, considering the COVID-19 situation in which we have a medical emergency and we need immediate solution, is that this is a near-term strategy. We can get immediate solution. It will save our time and money. And moreover, above all, the safety aspects of the drugs are well studied by the population because those drugs are time tested. So developing a new drug usually takes many years to decade or even longer, and nobody knows whether all these years would be a successful year or not. As SARS-CoV-2 is a newly discovered pathogen, so specific drugs have yet to be clinically approved. This is a diagram which is showing the traditional drug development way vis-a-vis the drug repurposing. So how it saves the time. Here you can say that we have to identify the therapeutic target for a disease, then develop potential drug candidates, identify the hits, design and synthesize them, and then we have to go for in vitro studies, then animal studies in vivo, then we have to do we have to go through the different phases of the clinical trial, and then the drug gets FDA approval. So it's a very time consuming process. Here you can see how we are skipping the all these barriers, and we have to only check, we have to do only few clinical trials, phase 2 and phase 3, and your drug is ready for the repurposed use. Now, how has been the experience with the drug repurposing? How successful it has been? So there are dozen of drugs which have been life-saving therapies, and they have been repurposed. Uh, for example, one very common drug, Viagra. Viagra was originally discovered for heart disease drug, but nowadays uh, it is used for erectile dysfunction. And I'm pretty sure hardly anybody, any general man, common man would be, who would be knowing that Viagra was originally discovered for heart disease. So gabapentin is another drug which was discovered for epilepsy, but now it is using, it has been repurposed for neuropathic pain, nerve pain. So especially the diabetic neuropathy patients, those who have problems, nerve related problems, they take these medicines. These are few drugs which have been listed, their original indications, I mean the diseases for which they were discovered, and the new indications, I mean how they have been repurposed successfully. You can see here amphotericin B. We all are from the biology background, and we know amphotericin B is a very well-known antifungal agent, but yes, now it has been repurposed for leishmaniasis. Aspirin was originally a drug which was anti-inflammatory and anti-analgesic nature, but now it is used as an antiplatelet, as a blood thinner. Similarly, I will not go into all the details. Methotrexate, I have shared with you all that methotrexate is a drug of choice. The moment you are detected with a rheumatoid arthritis problem, your serological tests are giving that you are RA positive, your ESR is reflecting the same, your CRP levels are reflecting the same. So the first medicine that comes to a doctor mind is methotrexate, depending on the condition. But yes, this is a drug of choice in case of rheumatoid arthritis. Now, we are talking about the drug repurposing, and if we don't talk about hydroxychloroquine, the whole discussion would be futile, because ever since the US President Mr. Trump has said that this is a fantastic drug with a very, and that too with a very little feedback. So it becomes very important and very very important to discuss the various aspects of the hydroxychloroquine. So hydroxychloroquine, we all know that it's a drug which was originally used for autoimmune disorders, rheumatoid arthritis, systemic lupus erythematosus, and in the countries where malaria is prevailing, it was used for as an anti-malarial drug, because it was having much better results in terms of side effects in comparison to the chloroquine. So hydroxychloroquine basically, uh, uh, it has multiple effects as it has been hypothesized. Let me tell you here, and I'll be telling you later on what are the several objections also which have been raised. So hydroxychloroquine, it has been hypothesized that it binds to ACE2 receptors expressed in the lung, so indicating that it may hamper the virus binding to the host cell. Similarly, another important function of the hydroxychloroquine is that it goes to the lysosomes, and then it raises the pH of the lysosome, and as we all know that the lysosomal pH is very low, highly acidic, so that highly acidic pH is changed and the pH raises, so that will affect the enzymatic functions of the lysosome, and proteolysis of the lysosome gets adversely affected. And this is one of the very crucial step in the virus, one of the essential step in the virus infection cycle. So thereby the hydroxychloroquine can check the virus progression, its replication. Another finding suggests that hydroxychloroquine has anti-inflammatory role. Yes, this is a well-established fact, because hydroxychloroquine was primarily used for autoimmune and anti-inflammatory disorders. So the hydroxychloroquine at the same time can check cytokine storm, also pro-inflammatory signals, which are governed by the IL-6, interleukin 6. So that's why hydroxychloroquine can be repurposed. But let me make you very clear that that none of us can decide whether one should take hydroxychloroquine or not. And in India, to the best of my knowledge, the hydroxychloroquine has been allowed only for the empiric use. Empiric use, we say that the use based on the experience, and that too to a very restricted population. That means it has been recommended for the use to asymptomatic households of the infected patients, asymptomatic healthcare professionals involved in the treatment of COVID-19 patients, and of course to the COVID-19 patients. But whether the medicine has to be given or not given, none of us can decide. It is the only doctor who is involved in the treatment who can decide. Another important medicine is nitazoxanide. Yes, this nitazoxanide has been reported that it upregulates the innate immunity interferon pathways, and thereby it helps us to check the virus infection. I must tell you here that hydroxychloroquine has several side effects too. And as Sanofi is a company which was primarily involved in selling the hydroxychloroquine, you all can check on their website. They have mentioned it that there's severe side effect. Hydroxychloroquine is and in Indian context, where there are so many people who are infected with who are suffering from the diabetes. So in that case, hydroxychloroquine has a side effect on the diabetic retinopathy. So the patients, those who are suffering from the diabetic retinopathy, if they may have a side effects of hydroxychloroquine. Similarly, some of the side effects of hydroxychloroquine on the kidneys has also been reported. So the hydroxychloroquine has several side effects also, and based on the studies, on which it was on which it was said that the hydroxychloroquine can be repurposed. It was a very preliminary kind of study. Earlier it was done on 42 patients. Out of those 42 patients, only 26 of them completed the treatment. So that was a very less number of population to substantiate any finding and concluding that yes, the hydroxychloroquine can work against the COVID-19 patients. Recently, one more finding has come on the almost around 1,000 patients, which has been done in France. But there are several reports. You browse these NCBI sites, and you go through several newspapers, New York Times, Guardian. They all have their speculations regarding this. Reason being that those 1,000 populations were taken. Ideally, the clinical trial should have been double-blind, randomized control trial. So the population, the inclusion and exclusion criteria which were taken, that is still debatable whether it was okay or not. So what is on the trial? So there are certain medicines which are which I have discussed with you also, chloroquine and hydroxychloroquine. They are on the trial for drug repurposing, and hydroxychloroquine along with the azithromycin have been proposed in certain countries. Remdesivir is an oligonucleotide analog which terminates the virus chain. Now, lopinavir and ritonavir is another drug. It is this combination is given for HIV treatment. You all would have heard that in Savai Mansingh Hospital, Jaipur, Indian doctors had given to some senior Italian patients, and those patients, they got recovered. From there, it came to picture lopinavir and ritonavir are the anti-protease inhibitors in case of HIV. People are trying to repurpose this also to repurpose in COVID-19 treatment. Now coming to the vaccines. So as we all know that the vaccines, we must select the epitopes which are the most up to date, and it's a time consuming, labor intensive, and huge amount of investment is involved in this process. So some of the vaccines which have been used in case of COVID-19, which are under the clinical trial, some of the vaccines which people are trying to explore is attenuated virus vaccine, protein based vaccines, virus like particle vaccines, DNA vaccines, and mRNA vaccines. Now attenuated virus vaccines, we all know attenuation means suppression. So the virus pathogenicity part has been suppressed, the antigenicity is retained, and then if these vaccines are given, we have a very good experience with them in terms of generating immune response. They generate a very good immune response because they mimics the natural condition, but at the same time they are very challenging, and there is always an apprehension that what if the virus reverts back to its original form, or what if the virus mutates to another more pathogenic form. So these are all apprehensions. Protein based vaccines, you all are familiar with this, so I'm not going to keeping uh, the maximum time limit uh, in mind. I'm not going to discuss these protein-based vaccines. You all are familiar with this. Yes, virus-like particle vaccines. They are also emerging as a very promising vaccine, at least in case of viruses, because in this case we can, as the term itself indicates, they are virus-like particles. That means they are not viruses, but certain structural proteins of different viruses can be clubbed in such a fashion that they can be used as a vaccine. So they also produce a very good immune response, and here since you can select multiple epitopes, so repeated clusters of the proteins you can have, and then you can get a wider range of immune response. DNA based vaccines, you all know that DNA vaccines, they are relatively very stable, that and that was one of the reason that they were selected. So DNA based vaccines are also under the in the clinical trial for COVID-19. DNA vaccine based vaccines have an advantage that we when these vaccines are delivered through some plasmid vectors or any vector, so they go inside our cells, and our cells produce their proteins, and our cells express those proteins on their surface, and then we get the immune response. So this is like mimicking the natural system very closely. We get very good immune response. Another important finding with the DNA based vaccines is that you can select the polarity of your immune system. Suppose these vaccines can be used even in case of allergic responses also. People, those who are suffering from the autoimmune disorders also. So why so? Because these vaccines, they can shift from Th1 to the Th2. Th1 is the pro-inflammatory side, and Th2 is the non-inflammatory side. So we can switch that also. So polarity of the T cells can also be manipulated by using DNA-based vaccines. But still the success rate of DNA vaccines in case of human trials is not very satisfactory. Yes, they are successful in veterinary usage, for veterinary usage, especially in case of horses, the Nil virus disease. In the Nil virus disease, the DNA based vaccines have been successfully used. The another kind of vaccine is mRNA based vaccine. These vaccines are emerging as a very promising vaccines, and for COVID-19 treatment also they have gone through a very successful, they are giving very promising results. mRNA based vaccines are far better than DNA based vaccines. So mRNA based vaccines can be a good choice. Another advantage of using mRNA based vaccine is that whatever they are doing, uh, whatever we are adding, the whole mRNA would be in the cytosol only. It can be translated there only. So there is no there nuclear involvement is not at all there. So chances of any side effect is very much minimized. Yeah, this is a typical timing that a vaccine takes place, and this is uh, these are the different types of vaccines which are under clinical trial. People are using for COVID-19 treatment. So 41 of these DNA vaccines, inactivated, live attenuated, non-replicating, and so on and so forth. Yeah, this all I have discussed with you, that what could be the different kinds of the vaccines which can be designed against the viruses. So let's skip this slide. Yeah, this is traditional paradigm. Multiple years takes place for a vaccine, but considering the outbreak paradigm, when we have a medical emergency, so we can shorten that development time. But even if we certain that, as per WHO reports, it will take minimum 18 months for a safe and effective vaccine. Now, these are the vaccine candidates, which have 105 vaccine candidates have crossed the clinical trial phase 1. Some of the promising emerging vaccines are these, as they have been named at the company's name, those who are leading this. Yeah. Now, how successful we are till date. So 66 programs are currently running in 3 different approaches. 7 are for repurposing drugs, 16 are for antibodies, people are working for neutralizing antibodies, and 43 for vaccines. Now, challenges in COVID-19 vaccination. So first of all, one thing which is very important, as I told it earlier also, the success rate is very low. So, and moreover, we have to take utmost precaution while designing a vaccine against a virus. So safe and effective vaccine is a big challenge in COVID-19 scenario. What if that the mutant versions are reported later on? Because the virus has traveled across the world, it is not confined to a particular region, so virus has been exposed to different environmental conditions, and in order to ensure its survival in the different changing scenarios, in the different changing host conditions, the virus has to switch on or switch off its genes. So considering all these facts, there are the chances are very high that there could be mutant versions also, and the same kind of findings have been reported that the vulnerability of the population is varying across the world. Some countries are very prone, some are less prone, and so on and so forth. In some cases, the immunity is not only and another problem is with these viruses that, for example, for flu virus, every season the flu virus changes, a new strain comes. So whatever immunity we are getting is for short term. And keeping in the mind that some unusual pattern has been observed in SARS-CoV-2 infection, that the patients which have been discharged, they were again found positive just after their discharge. And such type of unique, though very few, reports have been reported from Japan, China, and in India from Noida only, and in greater in our GIMS hospital. We are from the Greater Noida, so in one hospital, which is a nodal hospital, in their in Noida also, this type of 2 cases were reported. There is one organization, as I told you earlier, CEPI, Coalition for Epidemic Preparedness and Innovations. It indicates that the potential success rate is only 10% for vaccine candidates in 2019 COVID-19. Now, what should we do? What should be done? So, we should follow physical distancing but not the emotional distancing. We must have sympathy, empathy for those who are infected with this, and we should take care of all those who are around us who are deprived of the basic amenities. We must help them following all the government guidelines. As per WHO guidelines, there is a good news that 80% of the people are expected to improve without any hospitalization. So yes, this is a positive side that out of 100, 80% people, they don't get any severe kind of infections. That's a good news. Mind-body medicine. This I added from my side. I am a great believer of this. So mind-body medicine, it's one of the very promising emerging field in the field of medical psychology. The physiology and your physiological and biochemical parameters during the stress response have been observed to be changed. Now, stress has been linked with upregulation of several pro-inflammatory signals, and as per reports, pro-inflammatory signals and subsequent cytokine storm worsen the COVID-19 situation. As I told you that the cytokine storm is one of the main contributor in the COVID-19 death. Keeping that background in the mind, if we will become panic, if we will be under stress, so that would further worsen the problem. Several studies suggest that body heals faster and the immune system operates better when our body is in a state of relaxation. So we should relax, we should practice yoga, meditation, whatever we can do. Now the conclusion. since we are all under stress, so there is a general tendency that we want very easy and quick solutions, and we are searching for some miracle drugs. So yes, if we get it, nothing like that, but there has to be heedfulness, there has to one has to take the utmost precaution, and all the finding should be rationalized and not get overly excited by false positives. Better understanding of the drugs, especially their safety aspects, their toxicity endpoints, their organ toxicity, everything should be properly assessed, so that the drugs should not create more problems than they solve, and this aspect should be taken care of very carefully. Now let me give you the recap of complete presentation. There are three major findings, three major things which are emerging from this slide. One, that we should take utmost precaution in reporting the data, in getting the data related to the SARS-CoV-2. Second one, that the only and only immediate solution of this problem is drug repurposing. So we should explore more and more safer drugs which can be repurposed for this. And third one is, there is that the being panic won't solve the problem, rather being relaxed may help us. So these are some of the recent findings of our lab. They are our these Amresh Mishra and Yamni. They are my PhD student. They are very hardworking and sincere students. This is my lab. So thank you very much, and sorry for the inconvenience caused due to technical errors, technical problems. And I once again thank you all for joining us in this webinar. And there was a huge participation. We are really happy, and I again thank you all on behalf of School of Biotechnology Gautam Buddha University family. Stay home, stay safe, and stay relaxed. Thank you very much.